The next major shift that we're going to make in the course is to begin to examine not just the genetics of individual organisms, but genetics of populations. So a population is just a group of interbreeding organisms. So members of one species in a local area that can exchange alleles with one another. Let's consider a population that has two alleles within it. In laboratory crosses, oftentimes we'll talk about situations where there are more than one allele of a given locus, and in many populations, multiple alleles are present at certain loci. So here we'll use A again just as a way to represent a locus, and big A and little a as a way to represent alleles. Let's suppose in this population there are 360 individuals that are big A, 480 that are heterozygous, and 160 that are little a. What we can begin to ask now is to say, well, we have these two alleles in this population. What frequencies do they have? So we can ask the question, well, in this population, what's the frequency of big A? And likewise, what's the frequency of the little a allele? There's two different ways that we will do this, and I'll show you both ways. So the first way to add up alleles is simply to count them. Here we have 360 individuals that are big A homozygotes, so that's 360 individuals with two big A alleles each, which gives us, what's that, 720 big A alleles. And then we have an additional 480 that are present in the heterozygotes. So within this population as a whole, we have the sum of these two. So we have 1,200 big A alleles in this population. Then we can say, well, how many little a alleles do we have? Well, we have this 160. Those individuals have two little a alleles each. So that comes out to 320, plus this 480. The heterozygotes having one of each allele, so they count in both categories. And when we add those up, we can see that there are 800 of those. So in this population, we have 1,200 big A alleles and 800 little a alleles. So out of a total of 2,000 alleles for all the individuals. So 800 out of 2,000, that's a frequency of 0.4. And 1,200 out of 2,000 is a frequency of 0.6. So we would say the frequency of the big A allele is 0.6. 60% of the alleles in this population are big A, and the frequency of the little a allele is 0.4, so 40% of the alleles in this population are the little a allele. These two numbers should always add up to 1. Frequencies always add up to 1, and 0.6 and 0.4 indeed do add up to 1. Which is the totality of all the individuals in the population. So that's the first method. We'll now back up and I'll show you a second method that depends on the frequencies of different genotypes, which is in some cases easier to calculate. The way to calculate allele frequencies based on genotype frequencies is quite straightforward. It's mathematically equivalent to what we just did, but in some cases it's easier. We can say that the frequency of the big A allele is equal to the frequency of the big A individuals plus 1/2 the frequency of the heterozygotes. So in this case, now we need frequencies. So the total number of individuals in this population is 1,000, and we have 360 that are big A. So this frequency is 0.36, and then 1/2 of this frequency, 0.48 divided by 2, so 1/2 times 0.48, that's the same as 0.36 plus, work this out, 0.24. And you'll see that that comes up to that 0.6 that we just calculated for the frequency of big A. Likewise, the frequency of the little a allele is equal to the frequency of the homozygous little a plus 1/2 of the frequency of the heterozygote. We now have 0.16 for this, and as we just calculated, this is 1/2 of 0.48, So 0.24, and you'll see that that equals that 0.4 that we were just discussing using the previous method, and again those two add up to a total of 1. So this is a way to determine, based on genotypes or counting alleles, what the frequencies of these alleles are within this population. So as we just determined, in this population, the frequency of the big A allele is 0.6, and the frequency of the little a allele is 0.4. Now the question is, given a certain population, what sort of frequencies would we expect these different genotypes to have based on the allele frequencies that are present in the population? So now we need to start talking about the interplay between allele frequencies and the genotype frequencies. You can do this with probability, just as we've been doing all along. The question would be, okay, if the frequency of the big A allele is 0.6 and the frequency of the little a allele is 0.4, how often would we expect, say, the heterozygous class to form in this population, strictly based on these allele frequencies? Well, what does it take to be big A, little a? Well, we require a big A from the mother and a little a from the father, or we can have a little a from the mother and a big A from the father. Now the question is, well, what's the probability of getting a big A? Well, the probability here is the frequency of the allele. It's as if we're reaching into the population. If you imagine the population as sort of a jar of marbles, 60% of the marbles have a big A written on them, 40% of the marbles have a little a written on them. You reach in and you grab that first marble. What's the probability that it has a big A? Well, 60%. So with the females, the probability would be 0.6, and the probability would be 0.4 for the little a. 0.6 times 0.4 equals 0.24. Okay. Or it could be this way, 0.4 and 0.6. That also equals 0.24. And because we say or, we would add those two together, and 0.24 plus 0.24 equals 0.48, which you can see is the frequency that we would predict that this genotype category would have for these allele frequencies. Now, that makes certain assumptions. It assumes that all of these genotypes, that this genotype is not one that survives less than other genotypes or survives more. It assumes that breeding is random, that we're not having certain genotypes that are attracted to each other more than the other, so it's random. These genotypes don't select among themselves based on this locus, so breeding is random. We also expect what's called equal fitness, as we were just saying. Equal fitness means that they all survive and reproduce at the same frequency, that there's not sort of a genetic disadvantage to being little a versus heterozygous or homozygous. Okay? And also we'll assume things like no migration in or out of the population. So generation to generation, we're assuming that there's not large numbers of individuals coming into this population from elsewhere or leaving this population that shift the allele frequencies that we see here. We might ask the question, what would we expect the probability of being big A in this population? Well, what does it require? We need 0.6, we need a big A, from the mom, and we need a big A from dad. These would each have a frequency of 0.6. We're reaching into that marble jar again and pulling out a big A, and we need a second big A, and this is the only way that this category can be formed, so it would be the product of these two, or 0.36, which again is what we observe, just like we observed here. Okay, 480, probability of 0.48. The last one to do is the little a category. Again, we need a little a from mom and a little a from dad. There's no other way to make that category. And we know that it's 0.4 for each. And the product of 0.4 times 0.4 is equal to 0.16. which again is what we observe in this population. So this particular population, what we're saying is that the genotype frequencies are exactly what you would predict based on the genotype frequencies are exactly what you would predict based on the allele frequencies. The allele frequencies and the genotype frequencies are what you would predict from one another. Now, a population that is in this state has this setup where the allele frequencies can be used to directly predict the genotype frequencies that you observe in the population. This means that we have random mating, that we have equal fitness, everything is surviving at the same rate, reproducing at the same rate. It means there's no migration in or out of the population. It means that there's no large amounts of mutation which are kicking in new little a alleles, say, that sort of thing. Now, in reality, none of this is really happening, most likely. Most likely there is some mutation going on. There might be subtle differences in fitness. There might be, in a small population, not perfectly randomized mating. But in general, if a population fulfills these requirements, then its allele frequencies can be used to directly predict the genotype frequencies that you would see in the population. Such a population is said to be in what's called Hardy-Weinberg equilibrium, which we will abbreviate as HWE.